Welcome, everyone, to the Mind, Body, and Beyond podcast, where we talk about just that, the mind, the body, and beyond. I'm sharing different people, modalities, and products that are really beneficial to each of us as far as our physical body is concerned and then beyond. Now, there's a lot of talk these days about the microbiome, about being able to balance the microbiome, have a healthy microbiome, and the ripple effect of what that means. Now, there have been tests in the past, stool tests that not all doctors use. A lot of functional medicine, integrative doctors will, naturopaths, but it's something that can be costly, but can also not give you the full spectrum picture of a healthy microbiome. That is until Mark Driscoll, with the help of, I'm going to let you introduce more of the people behind it, with PacBio, created this product, GutID, which will show you a map of your microbiome. And ideally, it's healthy. And I'm going to share some charts of myself, my husband, and we may go into some other charts to discuss what a healthy microbiome looks like, the benefits of a healthy microbiome, and all of the implications this test has to be able to have a really strong basis of foundational information so that we can make choices in our daily lives, the foods that we eat, the activities that we engage in, the different supplementation that might be supportive, because a lot of us will read an article or hear something from a friend that really worked well for them, but then realize it's not actually ideal for their body, for their microbiome. It may be something that has an adverse effect that is a beneficial effect for someone else. So knowing your own microbiome and how to support it, I feel like is a fundamental foundational piece and the cornerstone of health. So with me is Mark Driscoll, the Chief Scientific Officer of Intas Bio, and Randy Erspamer, who is I believe the marketing, you tell me, Randy.
The head of business development for GutID.
There we go. So, and I'm going to let these two debonair gentlemen tell you a little bit about themselves. It's like getting the firsthand accounting of who they are and what they do. And so, Mark, let's start with you.
Great. Thanks, Amalia, for the opportunity to go on your podcast. This is great. First of all, I'm a scientist, and my motto is, if you can't see it, you can't study it. And that kind of tells you a little bit about me. My background is actually in next generation DNA sequencing, where my team and I, way back in the day, helped build the very first of those next generation DNA sequencers, which were used to sequence the human genome. And we did all kinds of fun things like help Svante Pabo sequence the Neanderthal and sequence a guy's DNA named James Watson of Watson and Crick fame. But one of the things that really fell out was the ability for the first time to see all the bacteria that make up your microbiome. And the company was really founded on the if you can't see it, you can't study it premise. And so the test that we put together is really, there's been a lot of information around gut bacteria, skin bacteria, mouth bacteria, and their effects on human health. But this ability to see it for the first time really drove us to found this company where at Intas Bio, we really delved into, on the medical side, started in the cancer space. And it's a little bit of a segue to how the test developed, but we're working on really trying to see how bacteria affect colorectal cancer with a guy named James Kinross at Imperial College London. And when we had the test running for that, he said, oh, by the way, since we can see all these gut bacteria, I have a lot of folks who don't have cancer who could use some insights when I'm seeing them about how their gut health is doing for things like IBS. So could I have a test that I could use to look for that? And we put this thing together, and we've been building on it until finally you have what you see in front of you. So that's kind of like my background, sequencing to bacteria to now gut health, sort of through the side link through cancer. And we still do have those projects going on, but GutID is really a way for you to see your microbiome, so you can study whether the changes you're making are actually having a beneficial or non-beneficial effect. And so with that, I'll hand it over to Randy to do an intro for himself.
Yeah, great. Thank you, Mark. And Amalia, likewise, thank you for having us on the podcast today. Super excited to share more about GutID and collaborate with you. So my name is Randy Erspamer. I'm the head of business development and run operations for Intis Biosciences and GutID primarily, which is our gut microbiome product under the Intis Bio umbrella. My background, I've worked in the biotechnology space most of my career. So I've worked for large biotech companies on the commercial side like Abbott Laboratories. Probably some of the most important work outside of Intis Bio that I've done is I've run antimicrobial resistance public health initiatives with the New York State Department of Public Health Wadsworth Center, with the Antimicrobial Laboratory Network under the CDC, and specifically tracking carbapenem-resistant Enterobacteriaceae and extended spectrum beta-lactamases, specifically in the spread of healthcare-acquired or nosocomial infections in large healthcare systems. And I've also worked for CLIA laboratories on the commercial side and operations side as well. But yeah, super excited to talk to you today about the GutID test and share some specific stories and case studies with you.
Wow. Yeah. So, I mean, basically the genesis or kind of the nexus point here is that I'm talking to two very smart guys who are very well versed in this space. So I feel like this is going to be a really powerful, informative podcast where people are going to walk away with some further information about what they can do for themselves. First of all, having the gut ID test as a great starting point. And I know when I met the two of you at a Biohackers Expo in Miami this winter, there was, the entire building was filled with people that were pioneers in their area of expertise, the areas that they really have had a personal connection to. Most everyone had a personal connection in some way to their work. And of that entire convention center, there were only a handful of people that I connected with and spoke to that really wowed me. And you guys were a couple of them, because of the fact that this, I had a stool test done with a doctor a few years ago when I was having some health issues, nothing that was really bothering me that much, but my husband was kind of harping on me to go and get checked out and, just see what's going on. So I went to this integrative physician who ran a gamut of tests, like a lot more than your normal GP would run. And the stool test was one of the tests that he ran. I tried to find it in order to really speak directly to what exactly he tested, and I could not find it. But I remember it was maybe 3 pages at the most, and it listed some different bacteria that I didn't understand or know anything about. Kind of the general consensus was stay away from gluten. I also did a food sensitivity test that said the same thing, along with a few other foods. But what I find really interesting about GutID is it almost, and you guys correct me if I'm wrong, it almost covers the basis for both, because it touches in on food sensitivities. It also touches in on, it goes deep on the different types of bacteria and shows you a visual picture. And I'm going to share my test results shortly so that I can have these two gentlemen speak about what they see and the different parts of my microbiome. And it's funny, my husband also did his, and we were kind of in this little mini competition, to see whose microbiome was better and healthier. And I thought for sure mine was going to kick his butt, but I was very, very mistaken. So, that was also kind of fun. So, I mean, as far as, families are concerned, it might be kind of interesting to see how each of you in the family, what your gut microbiome looks like, because that could really affect the meals that you make, the activities that you do, things like that. So it's like, to me, there's a bigger impact than knowing what bacteria is in your gut microbiome. It's really also a lived experience that you can take into consideration every day. And I love the fact that, this testing, which is so much more comprehensive, was around the same price as the stool test that I had that was just a few pages of, to me, kind of worthless information without a clinician or a physician that's able to read it, which is helpful. And I'm not knocking that, but for a lot of people, they might just be curious and want to know some basic things that they can do without having to go through all of that and be able to take some steps on their own. So I guess the question I have for you, Mark and Randy, I'm not sure which one of you would be better to answer this, was that part of the thought process in creating this, where it was like reachable for most people, attainable, and also, bringing in like all the different facets that would affect the health of a microbiome.
Yeah, I'll take that one first, and then we'll let Randy kind of riff on it. If you think about your gut microbiota, what you really are trying to get at is not necessarily for healthy people which bacteria are there precisely, but what we're looking for is a healthy ecosystem.
Mm-hmm.
And that's something that I think everybody can understand, because you can kind of tell if you think of ecosystems like a rainforest, right? So what do you think is a healthy rainforest? Well, first of all, there's got to be a lot of different things in the rainforest, right? So we know that the healthier ecosystems have lots of everything in it. And that's something that's really easy to see from the way we display the test, right? So you can tell when you look at your own test, like, does this look like a healthy ecosystem? The very first questions that you ask are, number one, are there a lot of things there? And a lot relative to what? Well, a lot relative to what we see in most people. And that's kind of the second part of this, is that we can, different people from different places, different diets, different backgrounds are going to have different bacteria in their gut. But the one thing everybody has in common is you can tell if there's hundreds of bacteria around those outer rings in our target plot, that's a healthy, diverse ecosystem. The second thing we really look at is a healthy ecosystem has an even distribution of whatever's in there, right? So if you think about it, when you look at a jungle, there'll be all kinds of trees, all kinds of plants, all kinds of animals, all kinds of birds, all kinds of everything. And one thing won't be everywhere, right? So if you have an ecosystem and it's full of 5,000 leopards and one bird, that's not a healthy ecosystem, right? And so that's the second thing we look at in this plot. And it's relatively easy in concept. You say, hey, are there a lot of things there? The more the better. Within reason.
Yes.
And then the second thing is, are they evenly distributed? So is something taking up a giant chunk of your microbiome? Well, that's not as good as if it took up kind of its normal space. So that's really where we start. And then the test itself breaks down into a bunch of, like, if there is something weird or wrong, we call out individual little parts of it to say, kind of like where it could be going weird or wrong. And this is a great example of kind of where we're looking at, are there hundreds of things around this outer ring? Not really, right? If you break it down, you've got something that's taking up almost half the space,
Yes.
something else that's taking up about a quarter of the space, and then everything else is kind of squished, right? That's not the healthiest ecosystem that you'd want. Now, you can take a deeper look at this and say, are any of these, like, quote, bad bacteria? No, not really. So it's one of those things where, there's no real big chunk of pathogens in here, but it's like more along the lines of a microbial imbalance where you've got, if we take a look at that big purple section called Bacteroidetes, there's a tan section called Firmicutes. Those two should make up almost all of your gut microbiome in an adult. The other one that we look at, that bifidobacterium, that one is also a healthy microbe, but it's one that you see in kids in high abundance. And if there's a lot of that in adults, it's a little odd, but it's not really concerning. But what you'd want to do is you'd want to make it so that big purple chunk of Bacteroidetes, which is mostly made up of one thing called Phocacola dorii, doesn't matter what it's named, it's taking up a giant chunk of space there. You want to push that down to a more reasonable level. And that bifidobacterium, get it down to a more reasonable chunk. And then you'd want to expand those tan firmicutes. Those are gram-positive bacteria that do a lot of heavy lifting in your gut in terms of digestion and gut health. You want to get those to be a bigger part of your microbiome. And so, you can do a bunch of things, diet, intervention, fiber, exercise, but then you can check back and see, hey, is what I'm doing actually working? And that's where my motto comes back to, if you can't see it, you can't study it, right? And so this is you, Amalia, studying yourself and what you're doing and saying, are the changes that I'm working on doing, getting me to a better place in terms of my score, my diversity, and that key metric that I mentioned called evenness, right? Where the P. dorii and the bifidobacterium are not very evenly distributed compared to the other organisms.
Yeah, and so it's interesting to hear you describing this, and this is a great example for people to see, what is happening, and then the other part of this. So it also interesting that there's a bacteria that's very unusual in adults. Like my mind goes to, is that based on what you're eating? Is that based on what I'm eating, or based on lifestyle, or that I didn't realize. Like, I don't know that was in the report necessarily. It may not be just kind of like a fun fact, not one that's, relevant to the, the path moving forward. But, interesting nonetheless. And then looking at the microbiome score as being kind of like the overarching view, of your microbiome as a whole. You can see in the, in the diagram that, in the chart, that where things are out of balance. And then also looking at, up in, if everyone looks up in the box at the top, there's a right underneath pathogens, kind of in the center, it says review. And there's 34 areas of review, 5 of which for me needed to be looked at. The worst was resistome, which is antibiotic resistant bacteria, if I'm not mistaken. And
yep, that's exactly right. And so some of that comes from P. dorii is known to be it's known to carry antibiotic resistance genes. And so since that's such a giant chunk of your gut, that is a commensal. It's a common bacterium that people have. But just having so much of that means that, if you were to take antibiotics, that would grow even bigger potentially, because it is more resistant to. So having that as a giant chunk of your gut already, if you were to go under some kind of antibiotic treatment, it would push that to even a bigger chunk of your gut. And we've seen people who have that at 80 or 90% of their overall microbiota. And so you want to avoid continuing down that pathway. And that's why I think resistome, in your case, sometimes you'll see people who have E. coli or Klebsiella or these other pathogens that carry a lot of antibiotic resistance. That's not the case here. You've just got this P. dorii that just happens to carry a small number of antibiotic resistance genes, but it's a big chunk of your gut. And so that's why that's the worst one. Now, even a healthy microbiome is going to have a worst thing, right? And so it's all relative. If you look at your top box, you can see your pathogens, 0.07%. That's fantastic. Your proteobacteria, 0.32%. That's amazing. And then fusobacteria, 0. That's exactly where you want that to be.
Nice.
So you're a great example of somebody who, could you have a better microbiome? Sure, that ecosystem could look better. Is there something like really wrong with it? Well, nothing in terms of pathogens, proteobacteria, fusobacteria. And so that's why it's like, it kind of gives you an idea that, you're an excellent candidate for, I would say, there's a number of things you could do to try to get that up. I had a huge overrepresentation of P. dorii, and I fixed that with fiber supplements. So, you can see if it works.
Well, and that's actually what I've been doing. I started with some organic psyllium husk, and what I found was that I did about a teaspoon, and it was too much. Like, it was too much for my body. So I brought that down to about a quarter of a teaspoon, and that seems to be kind of the sweet spot. I've been doing a lot of traveling and have some other things, so I haven't been super consistent with it, but it's one of those things that I intend to continue to integrate into my diet and lifestyle. And Randy, we haven't forgotten about you, because we definitely want to hear from you as well. But I just, as Mark was talking, I thought this would be a great time to introduce this, and so that people could see exactly what he was talking about. We're talking about the chart and the bacteria and the colors and the outer ring. So, and then one other note I want to make about this, and I'm going to give Randy a chance to share his thoughts too, is growing up, I had a fairly challenging childhood when it came to illness. I had very serious asthma as a child, pneumonia several times. I had viral meningitis. I was in the hospital. There was a lot of stuff happening within my body as a kid. And so I was on antibiotics a lot. And so I wonder, and maybe you don't know the answer to this question, if there may be a correlation between the antibiotic load that I had as a child, and then that the increase in whatever I can't see it on my screen to say what the bacteria is that you said that's unusual for an adult. I don't know if there would be a correlation to that or not.
It's always a possibility, right? We're talking about, everything in biology has to be viewed in the light of evolution, right? So we go back to Darwin and say, if you have bacteria in your gut and then you undergo antibiotic treatment, the ones that remain will be more resistant to the antibiotics. And so if you then undergo another antibiotic treatment, the ones that remain, and eventually you get to the point where just by kind of natural selection, you have reduced your sensitive bacteria and increased your resistant bacteria. And so we also know that once you have a profile, it tends to stick around. It takes a lot to move it around. And so it could well be, but it is something you can change. And so, with directed efforts, you can reduce that. And it might be, I mean, I know we want to get Randy in there too, but it might be a good time to show my report triptych because it does look very similar to what you have.
Oh, goody.
It kind of shows what you can do with, and once again, I am not an expert on microbiome treatment. I'm an expert on microbiome measurement. And those are as different as somebody who can build a race car versus somebody who can race a race car, right? So they're two different skill sets. And so when I had a terrible looking microbiome, I didn't try to fix it myself. I went to somebody who knows something about that, Elena Panzeri, our consultant, and said, what should I do? And she said, Mark, your diet is garbage. And so with that starting point, she said, you really need to change your diet so that you, and there you go. You can see that big blue starting point of P. dorii, which is the same thing, Amalia, that you've got.
Yeah.
It's antibiotic resistant. Once again, I think I'm alive because of antibiotics. But Elena said to me, your diet's trash. You need to do better. And so I said, well, like you, I know I travel, I'm busy. I know I won't make that kind of change. So I said, so what else can you do for me? And she said, oh, you're going to be difficult, right? And I said, yes, I am. Because, I know myself, and this is why, like, talking to somebody who's in the know can get, you personally the best outcome. Because she said, okay, if you're not going to make big dietary changes, the simplest thing you can do is just cram a whole lot of fiber down your gut. And so I said, that I can do because it's just like scooping that stuff up and down the hatch. And so unlike you, I'm not really sensitive to the psyllium husk. I can take just a ton of it. And so what I did was I just started taking it. I'll tell you, I was not a believer, right? So I had followed my microbiome at the starting point for 14 months. And it had not changed at all. My score was terrible, and it looked identical to what we're seeing there on the left for 14 months. And so I thought that there was very little we could do to push that around. But she said, no, take this fiber, go ahead, and we'll see what happens. And so by 4 weeks, things had started to change. And by 14 weeks, not 14 months, 14 weeks, you can see, like, where is that P. dorii? Well, it's kind of down on the bottom in that little blue slice down there. So when we're talking about an ecosystem, right, if you compare the left all the way at starting point to the right, where we're just like a snapshot, if you just didn't know anything about the bacteria at all and said, which is the healthiest ecosystem, you would say the one on the right, because there's a lot of things in that outer circle. They're evenly distributed. And if you look at, once again, that Firmicutes tan and Bacteroidetes purple, those should make up almost all of my microbiota, and they do. And they should be represented at about 50-50, and they are. And so it just made this set of changes that also raised my score up to a 92, which was way better than the 28 that I had before. And the last part of that was that I felt better. And so what do I mean by that? I'm getting older now, so I'm almost 60. And I thought, when you get to a certain age, the wheels start coming off the bus health-wise. And so I started having pretty regular gut pain after eating about 3 or 4 days a week, where I would have to stop what I was doing, lie down, and just kind of let it pass. And that kind of went away. And I didn't even notice that. I had just attributed that to my getting older, that I was going to have gut pain when I ate. But it was my wife who said, you know what, you're up and around and walking the dog and stuff after dinner instead of lying down, what has changed? And she noticed before I did that went away. So it is something where, I hadn't attributed my symptoms to my gut problem. But when the two went away, when the gut changed, those symptoms went away. Now, being a scientist, right? It's not like, did it work this once? The scientist asked, does this happen every time I do that? So I went off the psyllium husk, got back into gut problems, and then added it back again and came back into a healthy. So that's kind of the difference. Is most people would be happy with that having worked, but scientists always are like, does that happen all the time? So
And so, and
yeah.
Mark, was it only the psyllium husk that you changed, or did you also change your diet a little bit too?
Well, for me, I told Elena, I'm going to make this, or consultant, I'm going to make this even harder for you, because as a scientist, right, it's important to have, when you're doing an experiment, you only change one thing.
Okay.
And so she said, look, your diet is garbage. If you're only going to change one thing, this is the one thing you should change. And so it's helping my gut bacteria. I have not changed my diet significantly after that, just to see what happens once again, right? So I want to follow it for a long time and see, does this one thing for me make that change? Now, that's an N of one, so that worked for me. Doesn't always work for everybody, is what we found out. So,
Yeah.
some people can't tolerate psyllium. Some people need to do something else. Some people have like real problems, where there's pathogens in the gut and things that need to be addressed that are outside of just a normal dietary change. But yeah.
Okay.
That's
Yeah.
We see that all the time. Like, Amalia, you made a comment that you took less than the recommended dose for psyllium husk, and that worked for you. That's like one of the beautiful things of our report is it's a fantastic diagnostic tool to see the complete microbiome to the substrain level. But in addition, we add in very tailored recommendations to modulate your microbiota as the data shows. So it could either be prebiotic, probiotic, or potentially antibiotic therapy.
Yeah.
And the report will spell that out, and we'll get to the level of granularity that you need inulin as a type of fiber or a prebiotic to feed the microbiome, or that you need lactobacillus or Akkermansia as a probiotic for beneficial byproducts that those bacteria create as well. So the report will spell that out and provide that detail, and there's a reference table in the back as well. And then obviously with a clinician to guide you with the report, that'll be the most powerful tool to get you where you want your health goals to take you.
Yeah. Well, and I'm showing here, this is also my report, that shows the rest of the report, and it's fairly lengthy and just shows, the different scores, and, it does give you some recommendations of things that you can do, like you were talking about, Randy, that are really helpful, and to be able to support your gut health. We meandered all over the place, Randy, while you sat there very patiently and listened in. Is there anything that you would like to chime in beyond what you just did as it relates to what Mark and I were riffing on?
I mean, I think like higher level, Mark and the Intis Bio team have created a very phenomenal tool to tackle this interesting, unique challenge, right? That has really not been solved up until this point. We have this large polymicrobial community in our gut, and there's other tools or legacy tools out there like quantitative polymerase chain reaction or PCR, 16S from a sequencing perspective, metagenomics, metatranscriptomics. They all have their utility in healthcare. But when it comes to this large bacterial ecosystem that we have in our gut, none of them are perfect, right? They have different limitations like database dependence or the breadth of how deeply they can see, and short read sequencing has its limitations. So our tool is uniquely designed to be able to identify all bacteria in the gut and then drive these very tailored interventions to reach outcomes. And this has been established in peer-reviewed literature. Like very specifically a paper that was published 3 years ago in Microbial Genomics by Gehrig et al. Compared our Titan 1 technology to the technologies I just named, specifically looking at E. coli and the ability to detect E. coli in the gut. And what was found was that our underlying technology, which we call Titan 1, was able to identify 100% of the E. coli strains in this sample size. Meanwhile, the other technologies were only able to detect between 35 and 55% of the E. coli. So these tools are really very excellent at finding very unique bacteria or viruses or whatever the case may be. Like PCR, I think became a very common household name during COVID. Pretty great at looking for a very specific virus like SARS-CoV-2 or detecting HIV and quantitating it, but not a phenomenal tool for looking at a large polymicrobial community like we see in the gut. So that's like one of the unique things about the test is it's designed for this use case, and then driving an intervention off of it.
Right.
Now,
And Randy brings up a good point, right? So we've got a growing list of peer-reviewed publications where we're using the test. Like I said, James, with Dr. Kinross, we're looking at cancer, and we continue to look at that area. We have publications coming out with physicians who are looking at tracking infectious disease. So we work with Adam Matson. We've had the third of a trio of papers tracking infectious disease in the neonatal intensive care unit because he's interested in how are these bacteria getting past my defenses and how can I improve that? And so we're able to give him actionable information around that kind of thing where we know his infection control is amazing. So then how are the bacteria getting in? If you read those papers, you can figure it out. It gives him other knobs to turn in terms of keeping his kids healthy. So the very same technology that we're using to kind of scan your gut for the ecosystem, like Randy said, since we can identify these unknowns that are outside of any database, all the current technology, and I love that, right? I helped invent it, right? So all the engineering stuff, I'm very fond of it. But some of the Achilles heels are that a lot of these are just reliant on, if it's not been seen before, you can't see it. Because the first thing they do is they map all the data to what's known. And, for some of these folks, Randy's got a great example where this person had diarrhea because they had, for months, because they had a completely unknown mycoplasma that had taken up a giant chunk of her gut. And so every test said there's nothing there because they don't test for that. But kind of one of the unique aspects of this technology is since it sees everything, we can see if there's something there, it's at a huge amount, it's really messing with your ecosystem and nobody knows what it is. When it went away, the symptoms went away. So, you can draw your conclusions from that.
and so what happens in that case? So it's like, okay, so kind of to speak to what you were saying, Mark, like if you don't know that something exists, you can't really test for it. So with this, does it show like there's something that is somewhat unidentifiable that's taking up space in there, even though you may not know exactly what it is?
Yeah, Randy, if you want to pop up that slide,
Yeah, let me pull it up
we
here.
can discuss it. That's exactly what it does because what we do is, once again, our hypothesis is that we want a healthy gut ecosystem. Right. And so when this person, I think, Randy, you want to tell her story because she came to you and asked the question.
Yeah, happy to, Mark. So I met this woman at a biohacking conference in Miami, and she was walking by our booth, and she like literally grabbed my arm and was like, "Oh my God, what's different about your test?" And then she shared with me that she had been having chronic diarrhea for about 2 months, over a dozen times a day, waking up in the middle of the night with an urge to use the restroom. Obviously, a very horrible experience, and she believed it was onset from a food poisoning incident that she had encountered while traveling internationally. She also shared with me that she had taken 3 other gut microbiome tests. She had met with a few specialists to include a gastroenterologist, a functional medicine practitioner, her primary care physician as well. And they actually started to attribute her condition to stress and anxiety because all of the diagnostic tools that were used came back with nothing significant to report back. So I shared with her a little bit about our test and that we can see very deeply into the microbiome, and if it was related to her microbiota of her gut, that we would be able to see it. So she took the test, and this was her first result. And what you can see highlighted is this mycoplasma wedge. And to Mark's point, we are database independent. We do long read sequencing across 2 different genes in the bacterial genome, as well as a spacer region where mutations often occur. And it's highly variable in that region. And we read every single base pair across this operon in the bacterial genome. So There's basically no data loss when it comes from the DNA itself to going through our bioinformatics platform. So we see a perfect or near perfect copy of the bacterial genome. And then what we do is we basically look at the taxonomic hierarchy and we will find what the closest match is. So in this instance, we were highly confident that this was a mycoplasma that you can see here, but it was 5 novel strains of this mycoplasma that were within her gut microbiota. Interestingly, mycoplasma is this very unique life form under the kingdom of bacteria. It doesn't have a cell wall. It doesn't respond to antibiotic therapy. So for her specifically, if anyone would have given her empiric antibiotic therapy, it actually would have been antithetical to what she was trying to accomplish, because it would have wiped out her otherwise healthy diversity. She ate relatively healthy. She runs, she swims, she bikes, she shared with us. And you can see from like 5 o'clock or so around to 2 o'clock, she actually has very healthy diversity. It was just that this mycoplasma had taken up a good chunk of the relative abundance of microbiota in her gut. So what was recommended for her was prebiotic and probiotic therapy to basically expand upon one of the phyla of bacteria in her gut, which you see in the tan in this GutID plot, which are her firmicutes, to drive out the mycoplasma. And she did recently take a second test. After that intervention, she actually reached out to us 4 days later saying that it was the first day that she did not have diarrhea. She did admit, after we touched base with her a couple of times, that she had some up and down days, and it took her about a month and a half to get fully back to normal again. And then her second test, the mycoplasma, was fully eradicated. And last I spoke with her, she was back to her daily normal, if you will, before the food poisoning incident had occurred. So again, it highlights the power of the test. We were able to see something that other tests weren't able to see, drive a clinical intervention that obviously took this very horrible scenario of having to use the restroom a dozen plus times a day and bring her back to normal again.
Wow. Yeah. I mean, that to me speaks so directly to the power of this information to fundamentally improve someone's life. I mean, I felt so much compassion for her and what she must have been going through and trying to find solutions, just to be told like, oh, it's, you're crazy, or it's stress, or just like, we don't really have an answer for you. It's like, it's psychosomatic. I hear that a lot from people when they have something going on that isn't mainstream, or the doctor or physician or clinician doesn't have a sense of what it could be. And then they go directly to, oh, you're under stress, or it's anxiety, or whatever. And I feel like that's so unfair to people that really are struggling. And this is a great stopgap where they can, okay, let me see what's actually going on. To kind of piggyback on this and switch gears slightly, I'm curious what the two of you think about the connection between your mouth health, like your dental health, and then your gut microbiome. And the reason I asked that is I met a woman recently who does something called dental mapping, where she basically will identify different areas of your mouth. She said there's a connection between teeth that have been removed, like wisdom teeth, which is very common in the US to have your wisdom teeth removed. I don't know, Randy, if you want to stop sharing the screen for yeah, thanks. And Alzheimer's, the number of teeth that are removed has a connection to the higher rate of incidence of Alzheimer's. And then also just, like your gum health connecting to your gut health. Can you guys maybe speak about that, if you have some wisdom to share with us about those two connections? Because I feel like there is likely a kind of like a seesaw effect that might take place between the two.
Yeah, there's a lot of work going on with this right now. So just like on the extreme side, you talked about Alzheimer's, right? One of the discoverers of the Alzheimer's genes, Dr. Rudolf Tanzi, who's at Harvard, has a couple of papers out now saying that some of the plaque formation may well be the result of a normal or a unregulated response to microbial infection in the brain, right? So this is a guy who discovered the gene, right? Who's now pursuing potential links to bacteria. The other big deal is that there is a growing amount of literature showing that for things like colorectal cancer, pancreatic cancer, uh, prostate cancer, uh, even breast cancer. Leah Steensma out of Pepperdine University published a paper showing that some of these oral bacteria that are finding their way to other places in the body can be drivers of disease. In fact, some of the most recent publications around why there's an uptick in some of these cancers that are associated with older age, like colorectal cancer in youth, uh, may be driven by bacteria that are producing basically mutagens, right, in the gut. And so, if you're looking at something like colibactin, which is produced by E. coli, right, those E. coli are basically secreting a chemical which they use in microbial warfare, but in association with gut tissue, it drives the very AT-rich gene mutations that you see in the colon cancers. So some of these bacteria from your mouth, like Fusobacterium nucleatum, shown directly linked to, there was a publication maybe about 2 years that came out from the Fred Hutch, basically saying that Fusobacterium nucleatum animalis clade 2, which is a mouth bacterium, is a colon cancer seed.
Wow.
So we're looking at kind of for earlier interventions, for explaining like what are the environmental drivers of these cancers, right? Especially since we're seeing cancer earlier and earlier in younger people. We know that the human genome hasn't changed in the last generation. So by definition, human genetics can't be explaining the increase in cancers. Something is. And then when you take a look at these, just basically the environments in which we're getting better and better at manipulating the bacterial content of our bodies. And you have to take antibiotics, right? I mean, like I said earlier, I'm alive because of them, right? And so are many people. But then you can't also ignore the consequences, right? And so, sure, you clean up the disease using the antibiotics and that's all great. But then maybe you should pay attention to what the outcome of that is, because if the outcome is a highly imbalanced gut microbiota that has high antibiotic resistant, that really, before that, those drugs were available, that you didn't really see that in humans. I always ask the question, you have an intervention and then it works great, but then, you have to think about what could possibly go wrong, right?
Mm-hmm.
You've introduced this massive change. And now, since we know the gut microbiome can be fairly stable, you've introduced a massive permanent change. And sure, you've cured something which was life threatening, but then don't you want to try to put it back the way it was, right? Like if you fight a fire, right, you're spraying water in the building and you're breaking down doors and windows and that kind of stuff. And that's great. You put out the fire. Okay, super. But do you leave the building like that? No, you try to put it back together. And so that's kind of what this test is designed to let people do, is that if you've done something that was necessary, right, like a drug treatment, and you've wrecked the environment, get that ecosystem back to a healthy shape. And how do you know if you're doing it? Well, if you can't see it, you can't study it. So use something that lets you see it.
Yeah, Randy, do you want to chime in on that as well?
Yeah, 100%. I mean, to go further, like from the oral to the gut connection, I mean, Hippocrates said it best 2,000 years ago, and it's something that we've been further researching and learning about, is that all disease begins in the gut. And we've studied even beyond colorectal cancer and pancreatic cancer, but like the gut-brain axis is something that's incredibly interesting to me. And we have case studies on showing that individuals with an overabundance of certain bacteria, like Allostypes is a very common species of bacteria that creates an enzyme called tryptophanase, which basically creates lipopolysaccharides that causes an inflammatory response, starts the IL-6 pathway, can inhibit GABA and serotonin in the brain and contribute to anxiety, depression, other mental health disorders, or even neurodegenerative diseases like Alzheimer's or Parkinson's. And we have case studies on that, from optimizing athletic performance to just improving daily living. And gut health goes even further than mental health, right? It helps with immune response and metabolism. It can help with skin conditions like atopic dermatitis and eczema. And that's all something that we're studying with different collaborators on, and that peer-reviewed literature that is already out there shows. And we do actually report out on some of these disease states that I mentioned within the GutID report. We have a more complete one called the Complete Microbiome Assessment, and those disease states are specifically listed out with beneficial and detrimental bacteria that have been established in evidence-based peer-reviewed literature to the bacteria that contribute to these disease states because of the byproducts that they create in their metabolic reactions with different foods that you put into your body.
Yep. So we are, just for oral health, right? I mean, we know just with the links between oral health and heart disease, that having a healthy mouth is key to having a healthy life. And so those are areas we're actively exploring across all kinds of chronic disease states. And so stay tuned for papers coming out on this, in the relatively near future.
Yeah, that's really exciting to me because along with, as I mentioned before, the health conditions and illnesses that I had as a child, I also had a lot of activity in my mouth as far as orthodontic work and dental work. And so I had some sort of device in my mouth from the age of 3 to 18. And so that was a big part of, my youth. Then when I was, I believe in college, I was told that, oh, hey, you need orthognathic surgery, which is a very significant oral surgery after, 15, 16 years of very significant orthodontic care. So of course I said, no, thank you. I'm not interested in that. And so I did not have the surgery until I was in my 40s. And my surgeon said, this is a young person's surgery. I'm going to warn you, it's going to be a long haul. It's going to be a significant procedure. And we just want you to have all the information before you make this decision. And I had such pain in my jaw that I literally could not lay on my pillow on either side. And I was taking Advil a lot, which I knew was not a good thing. So, and I also had to get braces for a second time. So that was another kind of knock a girl when you're down. So I was going to the orthodontist with my kids, which they so loved that whole experience. And then I ended up having the surgery, which was a 9-hour surgery. I was in the hospital for 4 days. I wasn't able to eat. Using a syringe was the only way I could eat with liquefied food for 3 months. It was a year before I could. So I only share that story to illustrate, for me, I feel like there's such a direct connection between, and like you said, Mark, you saw my chart, not that bad, but there's definitely some things there that could be improved. And I feel very strongly that there's a connection between some of the I've got this hint of autoimmune stuff going on. I don't like to say, kind of own that I have whatever. It's there's some hints of autoimmune that I'm working with right now. And I feel like there's such a strong connection between the trauma of the oral and the dental piece of it, the microbiome, and how that affects us, in our bodies. Also knowing that the energy of trauma experiences or emotional experiences have an impact on us. This is the beyond part of the Mind, Body, and Beyond podcast, where, we have these little mini traumas that occur or larger traumas that occur, and there's an energy that's held within the body, and that can show up in different ways and in different places. The jaw being one of it, the gut being another place, wherever that may be. And I feel like this is such a great tool to be able to inform how to heal the body part of it, and then having someone that can help you walk through the emotional part of it, something that may have happened that you're holding on to that energy, and then that works in tandem. So, even talking about cancers, and I do believe, if I'm not mistaken, I read somewhere that there's you guys are working on or collaborating with others around addiction when it comes to, being able to draw the correlations between addiction and your gut. I too, Randy, am fascinated with the gut-brain axis. I think it's really interesting to see how those pieces fit together. I had a gentleman by the name of JP Erico on the podcast, and he has a device called Truvega, where he talks about the gut microbiome. And I thought, It's like, you guys need to know each other if you don't, because you would be a great partnership, with his device, and then being able to track, like literally track how vagal nerve stimulation is directly impacting your gut microbiome and creating fostering a really ideal environment, that is also impacting it. So, so many different tools and so many different approaches to really improving the state of our bodies, not just the gut microbiome, but how that affects all our life in general. I don't know if you guys want to speak on any of the different collaborations or other parts of the bringing the gut microbiome into a healthy balance. I mean, we've talked about a lot of different things, so I want to give you guys an opportunity to share anything else that you feel like would be beneficial.
Yeah, so as far as collaborations go, I'll let Mark speak to some of our academic collaborations and other biotech. But as far as on the B2B side, we work with concierge medicine, longevity clinics, gastroenterologists, functional medicine practitioners, nutritionists, dietitians across the board. And then GutID.com is also available direct to consumer as well for health conscious individuals. As we've discussed, the gut microbiome is core to overall health. Talking through all these different axes that I mentioned, from the gut brain, gut metabolism, gut immune, gut heart, and so on. So it goes much deeper than thinking like, oh, I'm suffering from constipation or diarrhea or IBS, IBD type of symptoms, but anyone that's interested in longevity, living long, but also health span, having healthy years as you age gracefully, the gut should be core to any program that you're considering, and GutID is available for clinicians and patients alike. And I'll let Mark speak to some of our academic collaborations that we're doing as well, and other opportunities.
Yeah, there's really too many to mention, but there's one that I think I've mentioned before. One of our basic questions that we're starting to try to answer is, where does your gut microbiome come from? And that happens immediately upon birth. And so I think I've mentioned Dr. Adam Matson's collaborations with us. We have kind of a 3-paper, if people want to get their feet wet in the literature, we have kind of a three-paper trilogy that we just completed. And the first was, how can we use the test to track infections at the substrain level? And he had a very basic question in that paper, which is, there are these bacterial pathogens that are making my kids sick. Are they the same ones time after time after time or not? And we're able to answer that question very definitively, is that they are not. So he had great infection control, and we could see the pathogens that were present today were not the ones that had been present in the past that he had archived. Okay, so the second question is, like, why do kids get sick? Because lots of kids have pathogens, but only a few of them get sick and get really in trouble. And so the second paper, we're able to show that it's okay if you have one pathogen. I mean, it's not okay, but it affects your health much less if these kids had 2 very closely related pathogens, but 2 of them. That was a sign that things were not going well. So that was paper number 2, like what's actually making the kids sick. And then the third paper we just published a few weeks ago is, where is it coming from? Because like I said, if you got excellent infection control in the NICU, where are these infections coming from? And it turns out that we're able to track these pathogens coming in through various methods outside. When the kids go outside the NICU, they might pick up in this case, it was in breast milk that we followed, that was pumped and came in, first established in the kid's gut as part of the gut microbiome, and then escaped into the blood and caused sepsis. And unfortunately, one of the twins died. So that gives also very specific, actionable information, right? It's like, clean the equipment better, right? Make sure those infectious organisms don't make their way into the milk and then into the kids. And so it's a great setup collaboration where, we're able to direct the folks responsible for infection control down the right pathway, because if they're always concerned about just transmission between kids and that never happens, you're not looking in the right area. So now you can look and say, okay, we've cleaned it, everything is clean inside the NICU. Now we have to look at where it's coming from outside, you can actually do things about that once you understand it. So these were a key set of papers that take advantage of what we do with GutID, which is identify substrain level unknown bacteria and follow them through, and then make actionable conclusions for, in this case, infectious disease control. But we have lots of collaborations like that.
Wow. Again, it's mind blowing to me how this test, though, has been like this launching off point for all these other inquiries and studies that have led to what sounds like significant strides in healing and health, not just for adults, but also for children and babies. I mean, that's a game changer, I would imagine, for that NICU, and then sharing that information with other hospitals potentially, and just being able
That's
to.
right. So there's a little summary at the end of this saying that the ability to see this for the first time at this level of resolution and with this throughput gives folks new tools for controlling infection in the most vulnerable patients. And that's the kind of thing where we're same technology, right, that we're using for looking at Amalia's gut and telling her their ecosystem is imbalanced. We can look at the infant's ecosystem and say it's not developing correctly, right? There are pathogens in this kid that when you get two of them, bad things can start to happen. So let's look at reducing them. And it gives a very concrete, you don't have to eliminate every pathogen, right? But when you're getting two of them, right, if you can reduce it enough that the kids never get two, you're going to be in a much better space. So it just gives you like, where do you point the big guns for control, right? If you're always focusing on the NICU, which is already great, you're not looking in the right spot. And it's always, if you can't see it, you can't study it, right? I mean,
That's right.
that's my motto.
That's right. I wanted to pull up my husband's chart as we wrap up here, because I feel like this might be a great way to like bring things a little bit full circle for folks, because the testing is very simple, and we both did this at home, and I followed the instructions very precisely because that's the kind of person I am. My husband's more of like, I'll just figure it out, and it's like fine. So I'm not busting him up a bit. I'm just like, I'm just poking him just a smidge. But what I found very interesting was that I thought for sure, since I followed all the directions, that my test was going to be better. And I was somewhat mistaken. And I just want to share his, just because what really struck me about his results was, let me pull this up really quickly, was that we had a similar score. However, he had 0 areas for review here. Yeah. And I was like, wow, like that's pretty impressive. Like even though his score wasn't super high, there was very little that he needed to change or do. Like, as we scroll down here where it gives you some of the, the scores of like what's high, what's low. I mean, there was some where there's like nothing, where it was just, he was right on the money. So I would just love for you to speak briefly, Mark, on, if somebody gets this test back, and it's super easy. Like I said, I follow the instructions. He didn't. Here's his results. Pretty darn good. And, it's a great place of inquiry. So for you, when like I saw this and I thought 0 places of review, that's great. Like you're doing really, really good. But maybe that's not the case. So could you share a little bit about that?
Yeah, I mean, so I'm not going to brag about my score, but I've got a 92, right? And so, and I started with a 24 or whatever the heck that was down there. So really, what that does is it says places for review. Is there anything that's really out of spec? And if you actually look at that, when we scrolled through that area down below where it had all the little bars and everything was in the green, a little bit, right down there, you can say, right, in terms of what you're going to change for this thing, you can see his evenness is probably, it's not spiked all the way to the right? And so, but if you look, like some of the differences that, like that Firmicutes to Bacteroides ratio, that's important, and that's kind of in the green. Fusobacteria pegged down to 0, resistome score low. That's because his primary Focia cola, if you scan up there, yours was P. dorii, and which contains antibiotic resistance markers, and his is P. vulgaris, which really doesn't. That's a lot more sensitive to antibiotics. And so you can see, but although, right, I mean, if you're comparing, look at his proteobacteria at 7.41%. That's much higher than yours. Now, the proteobacteria he's got is not particularly unusual or a bad one, so it's not going to detract from his score very much. And that's not a particularly high level, but yours was much, much less. So there, it just, you had a high resistome score just because of the predominant bacteria in your gut happened to be contain more antibiotic resistance genes. Now, does that mean there's no place to improve? And you can look right at this and say, hey, look, right, that P. vulgaris is taking up a pretty big chunk of the ecosystem. You want that to be a little bit decreased, right? And so, and look at the firmicutes, right? It's not exactly half of his gut. That could be better. So there are definite things that once you know where the mileposts are on this and say, okay, I'm looking at my firmicutes and Bacteroidetes ratio, they should be 50-50 and they should make up almost all the bacteria. It's like, it's about 50-50, but it doesn't make up almost all the bacteria. So that could be improved. And then we should have like everything evenly represented. And while it's pretty good, there's stuff in there that, it's a commensal bacterium. It's a good one, P. vulgaris, but it is overgrown. So, you can do better. Now, when I say that this is an emergency and you should go out and change everything, no, because things can also get worse.
Right. Well, and the great thing about this report as well is the fact that it tells you a few things that you can do. So then at least that way you've got a launching off point where you can start. And obviously, always have a physician or somebody that's helping you with your nutritional needs and making sure that you're not doing anything to harm yourself, of course. But this at least gives you some small things that you can change and tweak, potentially positively impacting your health overall.
And I think of this a little bit like, for a healthy person, it's like getting your cholesterol checked, right? And so, you might follow it over time and your doc will say, you're getting older, your cholesterol's ticking upward, you should really change your diet around a little bit. And then, it's the same test for a kid who's in their 20s versus somebody who's in their 60s. If it's a little bit high, right, you might take different actions depending on, if somebody who is 20 has a cholesterol level of somebody who's 60 on the edge, you're going to take different actions there, right? So it does matter, like not just what the test says, but the information around that, right? And so somebody also might say, my health score isn't that great. But if they've been working on it and it's way better than it was, that's better than, if it's decreasing rapidly. So there's a lot of context there. And so the recommendations in the report will give you things you can do, but that should always be viewed in the context of your individual, health needs and what you kind of got going on that about.
Yeah, that's a great point. That's very, very true. That's very true. Any final words as we're wrapping up here, gentlemen?
No, I just think testing your gut microbiome is a fundamental part of preventative care. Obviously, if you're having gut function issues or are concerned with some unexplained symptom, the answer could be in your gut. But honestly, we recommend testing at least on an annual basis with GutID because we really see it as a preventative test, right? You can see in your microbiota and with your score and these metrics that we've gone over on the conversation today, you can get ahead of different conditions, right? So proteobacteria can be linked to joint pain, which could be arthritis, rheumatoid, or otherwise. And if you start to see these bacteria developing in your gut, you can do something about it. You can't change your human genome, and we have exponentially more bacterial DNA in our body, but you can change your bacterial DNA and get ahead of these disease states and prevent yourself from going down this dangerous pathway earlier through dietary change, lifestyle change, supplement potentially, or potentially antibiotic therapy. So we may be a little biased, but there's a lot of other peer-reviewed literature out there as well that shows that gut health is core and fundamental. And I'm very fortunate to be here, and Mark invented this fantastic tool to be able to see deeper than ever into your gut microbiome and then make those interventions. So, yeah.
Yeah, and a little to Randy's point too, right? So just at some point, getting a baseline can be key. I have a relative who, she got sepsis and damn near died, and she's trying to get her gut health back to what it was before that because she was, a pretty healthy individual. It just came out of nowhere, but we have no idea what her gut looked like before that.
Yeah, that's a great point too. It can
Right.
give
So
you some.
just if she'd even had one test, we could say, here's what you had. Now, as you try to build it back, how close are you getting? We just have no idea.
Right.
We can work with it, but it's just getting a baseline is kind of a nice thing to have.
Yeah, absolutely. I agree. Both my husband and I plan on taking the test again and seeing where we are. The way I look at it is let's be consistent with some changes first, and then
Yes.
go back and see where we are, and and then also finding those like really simple ways. Like this might also be like where people have to find different strategies in their day to day. Like you were saying, Mark, like you knew that you weren't going to change your diet. That was going to be too traumatizing for you to do, too difficult. But you could add a little bit of fiber into your daily life, and you could do that when you were traveling. That was easy enough to do. So I think it's important that people find those places that they can take control of their healing process in ways that they're actually going to do. I mean, talking about it is one thing, but actually doing it and then following up to see, okay, are the changes that I'm making a difference, or do I need to actually take some extra steps to change some of the dietary pieces that I really didn't want to do, but I'm realizing I might need to do versus, something too extreme. I think, especially in this day and age, doctors are very prone to give a pill to solve things. And those pills, I think, are also contributing to the problems that we're having. So can we take a little bit of a different approach and then be able to see for ourselves how that approach is going? I think is also really empowering for people as well.
Absolutely.
Yeah.
Absolutely.
So, all right. Well, I want everyone to know that they can find more information about GutID. Mark and Randy, I appreciate both of you so much. It's gutid.com, if I'm not mistaken.
That's correct.
Okay. There's going to be a little discount code below if you're interested in purchasing that these gentlemen have been kind enough to share with me, so I can share that with you. And just one other footnote about the testing: it is very, very simple. It is very, very easy, and it's I think when people think about a stool test, it kind of grosses them out and skeeves them a little bit, and it really is probably one of the easiest things to do for any women out there or men out there who have kids. Like it's like not even as bad as changing a baby's diaper. So it's really not a big deal. I have faith everyone can do it. Super simple. So we thank you for being here, Mark and Randy. Again, I so appreciate your time, the work that you're doing, and the wisdom that you're sharing with the people you're collaborating with, the people that are going to listen to this and are, really making a difference in improving the lives of people on so many levels. That case study you shared, Randy, was really, really compelling. So, and I'm certain that there are people also with different cancers and addictions and mental health struggles that would really benefit from having some support. So thank you both so much. Thank you all for listening and being here with us today. We hope this has been helpful. If it has been, please do share it with someone you think may benefit, because as we've talked about on this show today as well, we learn from one another. Thanks for joining us. We'll see you soon.